Continuing with reproductive anatomy, let's go ahead and let's take a look at the female reproductive system in terms of its anatomical structures. So let's go ahead and take a look at what happens in terms of uh, oocyte maturation and follicle maturation leading to the ejection of the ovulate. And so what ends up happening is that we're looking at the principal changes taking place within the cortical region, in which we're going to have within the cortical region a development of follicles. These development of follicles will take place within the fetal maturation of the female. There will be a set number of follicles that will be allowed to develop beyond the primordial stage. Any of these cortical cells that do not develop into the primordial follicles during fetal development will become part of the secretory part of the ovary and be responsible for the production of hormones. These primary follicles will develop during puberty. And then as we get secondary signals coming from a combination of estrogen, FSH, and LH, we get a expansion of the primary follicle and the terminus of the first wave of meiotic division that takes place. We get an expansion of the cells surrounding the oocyte, the granulosal cells. This expansion of cells around the oocyte leads to the formation of the secondary follicle contained within the antrum surrounded by the zona pellucidea. We get a separation of granulosal cells during this secondary follicular development. This is where we will get the 3 layers of cells developing within the ovulate, which will be released. We then continue to have maturation of this secondary follicle into what's referred to as the vestibular follicle. The vestibular follicle is going to have an expanding antrum and a condensing of the granulosal cells surrounding the zona pellucidea, which is going to surround the secondary oocyte that is developing within the secondary follicle here, or the vestibular follicle. This set of granulosal cells will form what's referred to as the corona radiata. During this vestibular follicular phase, the antrum will be starting to fill with fluid, and what's going to do is it's going to start to build up hydrostatic pressure. As the hydrostatic pressure begins to increase, it becomes larger than what the tunica albicans or the germinal epithelial tissue surrounding the ovary is able to withstand. This leads to a set of muscular contractions within the ovary that pulls on the ovarian ligament and suspensor ligament that arranges the ovary so that it will line up with the infundibulum of the fallopian tube to allow for the fimbriae to sweep the ovulate into the fallopian tube. Upon release of or ejection of the ovulate, the remaining antrum starts to collapse, and all of the granulosal cells which have been producing estrogen and progesterone continue to produce estrogen and progesterone in what's referred to as the corpus luteum. This corpus luteum will remain as long as progesterone and estrogen are necessary to be produced. If fertilization takes place, if there is a activation sequence following interaction of the sperm and the ovulate, intrauterine signals signal the ovary to keep producing progesterone and estrogen to keep the corpus luteum intact. If those signals do not take place, the corpus luteum stops producing, the granulosal cells stop releasing estrogen and progesterone, and it will collapse onto itself because there's no longer any fluids within the anteromal region to keep the corpus luteum intact, and it will form what's referred to as the corpus albicans. If we look at what's being released, this is where people will claim that, oh, this is the egg. There is no egg. You have an ovulate, or females have an ovulate, Inside the ovulate, we are going to have the oocyte. This oocyte, when it is ovulated, is a diploid cell. It has not finished the full meiotic division. The full meiotic division will not terminate until it interacts with the sperm, leading to the formation of the secondary polar body in a fertilized state. Surrounding the oocyte will be the zona pellucidea and the corona radiata. These are protective cell layers. The protective cell layers are there to minimize the amount of interactions that a sperm has with the oocyte. And upon interactions between sperm and oocyte, it will ensure that one and only one sperm will be able to interact with the oocyte, leading to fertilization and the formation of the zygote. So we start with the ovulation. The ovulation will involve with realignment of the ovary towards the infundibulum of the fimbriae, and we'll start getting peristaltic contraction of the fimbriae, along with celiac contraction of the endometrium within the fallopian tube to start to sweep the ovulate towards the fallopian tube itself. We will then get ciliary contractions and peristaltic contraction within the fallopian tube, propelling the ovulate towards the uterine horn. This is the region where we will see interaction between the ovulate and the sperm, leading to possible fertilization. We will then get implantation, hopefully within the fundic and body region of the uterus, due to expression of receptors for implantation. Any receptor for implantation outside this region towards the fallopian tube can lead to ectopic pregnancies. Ectopic pregnancies, regardless of what you may have been told, cannot be reimplanted. So the last of the anatomical structures to look at are more for post-natal care of the infant, and that will be the mammary gland. The mammary gland sits anteriorly and superficially to the pectoral muscles, pectoralis major, pectoralis minor, as well as the intercostals and underlying costal bones. It's going to be held in place by ligamented structures in which we look at the various lobes. The lobes are held in place by suspensory ligaments. Suspensory ligaments are interwoven with fat pads and run contiguous with the deep fascia that is in line with the pectoral muscles. The suspensory ligaments of the lobes ensure that the lobes themselves are angled correctly for lactaceous sinus to allow for suckling to occur. Inside of each of the lobes, we will have a lobule. The lobules are going to be arranged with different types of secretory and myoepithelial cells. In the center of the lobules and heading towards the lactaceous sinus, we have the alveola. The alveola will lead to the lactaceous ducts. The lactaceous ducts will lead to lactaceous sinuses. The lactaceous sinuses will exit the mammary gland through the nipple. The nipple is surrounded by a cluster of mechanoreceptors and glandular tissue known as the areola. If we're to look at the lobule tissue itself, within the lobule tissues, we're going to have a collection of differential secretory cells. These differential secretory cells are referred to as the luminal epithelial cells. These luminal epithelial cells are going to be responsible for the production of lactations, quote unquote, breast milk. Surrounding these epithelial cells is a secondary set of epithelial cells referred to as the myoepithelial cells. The myoepithelial cells are specialized smooth muscle. These smooth muscle, under pressure from and stimulation by oxytocin and suckling, will lead to contraction. The contraction will slowly reduce the volume of the sphere within the lobule.
propelling the secretions within the cells into the lumen. Now, these cells within the epithelial will constantly be producing lactation under stimulus of estrogen and prolactin. The production of milk is required for prolactin and estrogen. It is not necessarily because a female carried an infant to term. As long as prolactin is high and estrogen is high, these epithelial cells will be able to produce. The age or the nature of interaction between the infant and the female does not dictate whether or not these cells will be able to produce and release lactation. These cells are sensitive to estrogen, and their sensitivity to estrogen can be impacted based off of suckling behaviors. There is some evidence to indicate that females who are susceptible to breast cancers due to genetic predispositions who do not suckle have an increased likelihood for developing estrogen sensitive or estrogen derived breast cancer within the luminal tissue of the gland itself.